everyone, welcome to the course on medical biomaterials. In this class, we will again talk about biological responses and slowly move towards in vivo and animal studies. Any biomaterial before it goes into clinical trials has to be tested on animals. So, we will start talking about animals also soon. Large number of animals are used like rabbits, mouse, rats, going right up to dog, sheep and so on. So, let us continue some more on the biological response. So, as soon as the biomaterial is placed inside the body, I said in the previous class there is going to be a blood material interaction leading to the activation of the coagulation pathway and then the activation of the complement pathway. Both of them are part of the immune response system. Then what happens is things start happening. We have the inflammation, that is part of the tissue response, okay. Acute inflammation, the chronic inflammation, then the tissue formation, foreign body interactions and fibrous encapsulation of the material and so on actually. So, a lot of tissue response starts happening here. So, we will look at it slightly in more detail. So, when there is an inflammation, there are a lot of wound healing response, because when the surgeon tries to open the host body and then place a biomaterial, obviously it is like a wound. So, the wound healing processes start taking place because the surgeon has created an injury, okay, because of the biomaterial implantation. So, there is going to be inflammatory cell infiltration. So, a lot of cells like neutrophils, PMNs, monocytes, lymphocytes start going towards it. Then there is going to be edema, vascular leakage. So, all these start happening actually, okay. So, the biomaterial is placed inside. So, there is a plasma protein adsorption, then we have a provisional matrix formation, monocyte addition to the biomaterial, which here you have the complement and coagulation, which we talked in detail. Then we have the macrophage differentiation, and macrophage plays a very important role in forming a lot of foreign body giant cells and trying to engulf or encapsulate the biomaterial, okay. So, they are all interrelated, as you can see here. We have the acute inflammation, chronic inflammation, then the tissues are formed here and your biomaterial gets completely fibrous capsule formation. So, both the things happen simultaneously when you have the inflammation healing process. Now, these are some of the inflammatory cells, as you can see in this picture here, and these are granulation tissues. As you can see, these tissues are formed around the biomaterial and they try to completely encapsulate your fibers, okay. And as you can see, these are some of those monocytes which are formed near the site of inflammation or near the site of where the biomaterial is placed, okay. So, these are part of the inflammation and wound healing process, okay. So, a lot of different types of cells are formed here, and then finally, if the biomaterial is inert, it can completely get encapsulated. Of course, as I said, if the biomaterial is toxic or the exudant or toxic, then you could have cell death happening there. These are some again pictures of foreign body giant cell formation. As you can see here, this is called a foreign body giant formation. So, different types of cells are formed. I showed this picture a long time back: lymphocytes, plasma cells, macrophages, fibroblasts, neutrophils, eosinophils. So, all these are formed here in this particular step here, okay. So, look at this, there is an experiment. So, when we use the mouse raw 264.7 cells, what happens when it is in contact with the biomaterial like polyester, then this is a polyester, this is a modified polyester. So, what are the inflammatory response when the mouse cells are in contact with these polyester? This figure tells you the expressions of two important transcriptions which are involved in inflammation. One is called the TNF alpha, the other one is called IL-1 beta, Okay. These two are markers which tells you if there is a inflammation. As you can see in the control, they are very low, okay, amount of the level of TNF alpha as well as IL-1 beta. But as soon as they come in contact with the biomaterial made up of polyester here, so you can see both of them have gone up tremendously by almost 12-fold, whereas when they are in the control, that means there is no contact with biomaterial, it is almost like 1-fold. Okay, so it is shot up. So what we do? We do some modifications to the polyester. So you are able to bring it down from almost 12-fold to 4-fold. So a 3-fold big reduction in the inflammatory response, which you have achieved by modifying the surface, because surface modification helps you to reduce the inflammatory response of the biomaterial to this particular cell line. So, these type of experiments have to be done in your lab before you take this biomaterial further for animal studies. So, these are called in vitro cell line based studies. So, we are using this particular cell line, you can test other muscle cell lines and so on. Okay. And as you can see, as soon as they come in contact with the polyester, there is a big increase in the inflammatory markers. So, most of the in vitro studies with cell lines focus towards these two particular inflammatory markers. One is called the TNF alpha, the other one is called the IL-1 beta. Okay. So, they look at the transcription levels of these two markers, okay, and then you will be able to tell whether the biomaterial is creating a inflammatory response. So, we do some surface modification to this biomaterial, and as you can see they come down dramatically, but still it is higher than the control, but it has come down dramatically. Okay. And so, how do you do these experiments? There is something called real time PCR, okay, polymerase chain reaction. This is an instrument which can tell you what happens to various genes. So, we can focus on a particular gene and then tell whether those gene levels are up when they are in contact with the material and so on. So, it tells you the mechanism. This is called a mechanistic study. So, we are able to tell there is a inflammatory response because these particular two genes, mainly TNF alpha and IL-1 beta, have gone up by almost from 1-fold to 12 times increase, okay, 12 times increase, and this one is 10 times increase. But when you do a surface modification, you are able to bring it down to only 3 times increase in TNF and 2 times increase in IL-1 beta. So, do you understand? So, this type of experimental studies are very essential to understand the inflammatory responses these materials create, and then you can modify the surface of the material so that the inflammatory response is dramatically reduced. So, we may be able to reduce it still further by another type of modification. Okay. We will not go too much into that as of now on surface modifications, but surface modification of biomaterial is a very important topic. You can do it through different approaches, plasma bombarding, UV radiation, immobilizing or coating antibacterial material. Yesterday I talked about coating heparin. When heparin is coated, it prevents the complement activation or even platelet activation, immobilizing enzymes and so on. There are so many different ways of modifying the surface so that we can reduce the blood coagulation, we can prevent the activation of platelets, we can reduce the inflammatory responses and so on actually. Okay. So, this type of in vitro study is very important before you actually go into in vivo animal studies. Okay. Okay. So, biocompatibility basically measure of the magnitude and duration of the adverse alterations. Okay. It is not only how bad the response is, but how long it lasts in the hemostatic system in the host. Okay. It should not create any adverse biological reactions, At the same time, the medical device should perform as intended. It should not present any significant harm to the patient. Okay. So, these are all necessary when you want to say the material is biocompatible. So, many assays are there for assessing biocompatibility. I have been talking about it in previous classes also. So, sort of we will summarize them. There are in vitro assays, that means we can do it in lab. There are in vivo assays where you do it in animals. So, in vitro assays, I mentioned it long time back, cytotoxicity, toxicity to cells, cell adhesion, that means are the cells adhering properly, and then are they proliferating properly. Is there a problem in adhering? Is there a reduction in the proliferation? That means cells are dying, cells are not growing. Okay? That is called cytotoxicity. So, we are looking at cell death, altered membrane permeability. So, as the membrane become very porous, so some of the enzymes inside which are important come out at the cellular level. So, we can monitor even that also. We can look at whether membrane has become permeable using different types of dyes and looking under a microscope. So, we can have fluorescent dyes, and if the membrane is permeable, then those dyes may go inside, and then we can look at it using a microscope, fluorescence microscope. Okay. We can also look at specific cells. There are so many different types of cells we can look at. We can look at like I showed you in the previous case, mouse cells. We can look at human L6 muscle cells. We can look at bone marrow cells, so on. So, we can look at the cell culture on specific cells. Okay. So, then we can look at whether cells are adhering properly, because that is also very important. Adhesion is very important, so that the cells can start proliferating. Okay. So, the adhesion depends on the properties of the biomaterial, like surface charge, hydrophilicity, hydrophobicity, what is the surface roughness, and so on. So, we can do that sort of cell adhesion studies experimentally in the lab, okay, in vitro based. For example, I just want to show you an example of how to do. What we did was we have a biomaterial made where we use different types of beta glucan. Glucons are produced by bacteria. These are all bacterial glucons. So, they are generally some of them are water soluble. In this particular case, this is water soluble, they are very biocompatible. This is carrageenan. So, we prepared 4 types of gels, one with the no glucan, only carrageenan, okay, beta glucan it is called. Beta glucans are also supposed to be immunomodulating properties, and this gel is prepared only with 100% carrageenan. This gel is prepared with 5% beta glucan and 95% carrageenan. This gel 25 is prepared with 25% weight percent and 75% carrageenan. This gel is prepared 50-50. So, 4 different gels with the increasing beta glucan. As I said, beta glucans are produced by bacteria. They are biocompatible, they are water soluble, some of them are water soluble. In this particular case, it is water soluble, molecular weight about 1500 Daltons, and then we grow cells on them. Okay. We grow this particular cell 3T3 Swiss fibroblast. Okay. So, look at this on gel 0, that means there is no beta glucan, only carrageenan. Cells are not adhering properly, and the cells have to look spindle shaped, whereas they all look spherical shaped. Obviously, this particular material surface is not very conducive. So, it is toxic to the cells, 3T3 cells. Now, look here, as we keep adding beta glucan 5%, 25%, 50%, you can see cells are becoming spindle shaped. This is the correct morphology of the cells. Okay, they are becoming spindle shaped here, very nice, very nice. So, by adding more beta glucan, which is biocompatible, you are allowing the cells to adhere as well as grow in a proper shape, which is the spindle shape. Whereas, if you look here, when there is no beta glucan, they are spherical. Obviously, this is not the correct. So, addition of beta glucan is improving the biocompatibility of the material. So, you can do this type of studies in vitro in the lab. So, the amount of adhered cells also increases as you increase the beta glucan, and they also achieve their spindle shaped morphology. You can see this, right. Okay. So, these are called adherence test, which is carried out in vitro. We can look at any type of cells, okay, and then we grow the cells, and then we can look at them under a microscope. So, it is quite easy actually. Okay. Okay. You can do another study that is called scratch assay. This is again an in vitro. In vitro means you can do it in the lab. The scratch assay tells you whether the cells are migrating. That means they grow and start migrating. If the surface is very biocompatible, cells will nicely migrate. The surface is not biocompatible, it is toxic, then the cell migration will be slow or completely retarded. Okay. Say same material, we took 4 different surfaces. This has got 100% carrageenan, this gel 5 has got 5% beta glucan, 95% carrageenan, this gel 25 has 25% beta glucan, 75% carrageenan, this gel has 50-50 beta glucan and carrageenan. So, what you do is we grow the cells, okay, for 24 hours, and then we create artificially a scratch. Scratch means we make a mark with a very fine needle, and then we start seeing whether the cells start migrating. So, slowly the scratch should completely disappear. So, this is 0 time, okay, this is 0 time on gel 0, gel 5, gel 25, gel 50. We created a scratch, and then after 24 hours we are monitoring the scratch. This is a color image, we have put in a dye. So, you can see only carrageenan is there, the gap is very large, whereas the gel 50, which is 50-50 carrageenan beta glucan, the gap is very small. That means cell has proliferated, migrated and started reducing the scratch. Okay. That means the material is very conducive for the cell growth. It is a very interesting experiment which you can do in vitro, and this experiment also tells you we can use this type of material because they help the cells to proliferate, migrate, which are very important if you want to have a very highly biocompatible biomaterial. Whereas this particular gel 0, which is 100% carrageenan, look at this, even after 24 hours the migration is very poor. That means it is slightly toxic. Okay. As you keep increasing the beta glucan, we can see the migration, the proliferation is very good. Okay. This is shown numerically here. Okay. So, gel 0 migration, if you take it as 1, and the 25 and 50, the migration goes to almost 2 times. Okay. So, double the rate at which cells are migrating. So, these assays, the in vitro assays I talked about in the previous slide, the adhesion and the morphology development and the scratch assay, all these assays are very useful to determine whether a biomaterial is conducive to the cells. These are experiments done in vitro, that means in the lab. So, we can use different types of cells, as I said, here I am showing you 3T3, we can use raw cells, mouse and so on actually. So, I showed you a lot of different in vitro based experiments, monitoring the inflammatory markers like TNF alpha or IL-1 beta, looking at cell adhesion, proliferation, looking at cell migration using this scratch assay. All these are experiments which we carry out in the lab to prove or disprove whether a material is biocompatible or non-cytotoxic. Then we can modify the surface and again see whether the cytotoxicity is reduced. Finally, once we are satisfied, we can go to animal studies. Okay. So, the in vitro cytotoxicity assays is, we take the material polymer for example, we see whether the extracts from the polymer are going to create toxicity. Okay. This is very useful especially in dental implants. For example, dental PMMA, polymethyl methacrylate is used widely. How do they do? they take methacrylic acid and it is polymerized using UV. So, most of the methacrylic acid is becoming polymer, most methyl methacrylic acid becomes polymeric methyl methacrylate, but little bit of monomer is left behind, little bit, which may slowly leach out over a period of very long time. So, is these leachants toxic? So, this type of assay is very useful. Okay. So, what we do is we extract from the polymer and use that extracted solution and see whether the cells grow in that, whether the cells die in that, and so on actually. For example, you are using amalgam, lot of amalgam based materials are used in dental fillings. So, they can slowly leach out over a period of years. So, are they toxic? So, this particular assay is very useful. So, you take out the samples, extracted liquid from the material and then test whether that liquid is toxic to the cells. Direct contact. So, what do? You incubate the material, biomaterial with the cell lines, and that is called direct contact. Third approach is called agar diffusion. So, what you do is we have cells nicely growing. So, we add a thin layer of agar solution on top. Agar is used for growth of cells, bacteria and so on. Then once the agar solidifies, your biomaterial is added to this, okay, and then you check. So, if the biomaterial is toxic, the cells will not grow closer to the biomaterial, but if the biomaterial is not toxic, cells will completely grow even near the biomaterial and start engulfing it practically. Okay. That is called the agar diffusion. So, this method is almost like your cell attachment and proliferation and migration, okay, whereas these two methods are like cell proliferation or cell death. So, these are in vitro methods, these are more qualitative because we cannot get a quantitative number, but there are quantitative methods are also there. I talked about it long time back. If you remember, we used to do MTT assay. Do you remember? In our old class we did, otherwise you can go back and check. It tells you the cell metabolic activity. So, we can calculate in percentage with respect to control how many cells are viable cells are there. Okay. We can say 90% viable cells, 80% viable cells. Something called MTS assay, it is again looking at cell proliferation, percentage of cells alive when it is with the biomaterial with respect to the control where we do not have a biomaterial. Then we have the LDH assay because there is this particular enzyme called lactate dehydrogenase, which is present inside the active cells or cells which have a good metabolic activity. If the cells die because of necrosis, then the membrane becomes damaged. So, the membrane because of the reduction in membrane permeability, this particular enzyme comes out into the solution, and the measure of the amount of this is a measure of the cell that is died because of necrosis or membrane damage. Another is nuclear condensation in apoptotic cells. So, if the cells die naturally, which is called apoptotic, and we add some dye, we can see the nuclear condensation under a fluorescent microscope, and we can tell whether it has died because of apoptotic. If it has died because of unnatural causes like necrosis, there will not be condensation of the nucleus will be spread all over the place, so we can tell the necrosis. Okay. These are called quantitative assays. I talked about it some time back, and these are the qualitative assays. Okay. So, all these assays are very useful in vitro to understand the cytotoxicity. Then hemocompatibility, that is the blood related compatibility. You want to know whether the effect of implant on blood and blood components. Again, I talked about it long time back because the implants can cause disruption of blood cells, that is called hemolysis, or they can activate the coagulation pathway, okay, leading to blood clots. If you remember, we talked about fibrin and then complement pathway. So, the implants can activate the complement pathway, which leads to several proteins getting activated. So, the ISO standards are there, which talks about 5 different test categories: thrombosis, coagulation, platelet, hematology, immunology. So, one can do all these tests to understand whether the material is biocompatible, okay. And there are 2 levels of tests, the basic level and the advanced level, okay. So, again the hemocompatibility depends on the material characteristics, fluid mechanics of the implant device, coagulability of the blood, okay. So, if somebody is having cardiovascular problem and he or she is given blood thinning like warfarin or even aspirin, so the coagulation does not happen so easily, okay, whereas a normal person the coagulation may happen easily, okay. So, hemocompatibility also depends on that, okay. So, I also talked about how to reduce the activation of the coagulation as well as the complement pathways, okay. There are coatings which can reduce that, we talked about it some time back, and we also said there are certain synthetic polymers which are very hemocompatible, okay, and which does not activate these coagulation and complement pathways. So, one can think about using those biomaterials, okay, or think about coating the biomaterial, okay, which will reduce the activation of these particular 2 pathways, okay, that is called hemocompatibility. So, there is a hemolysis assay, okay, for all materials except those that come in contact of only intact skin and mucous membrane. So, this assay tells you how much of the red blood cells are damaged with respect to the control, okay. So, you also have the coagulation assay. So, it tells you the effect of the material on the coagulation time. One is called the prothrombin time assay, other is called the partial thromboplastin time assay, okay. Both these assays are quantitative, so it tells you the effect of the biomaterial on the coagulation with respect to the control. Control means without the biomaterial, okay. Then there is again assays for complement activation. So, again as I said the complement activation with respect to the circulatory blood, okay. This is also an in vitro assay. So, it determines the complement activation in human plasma as a result of exposure of the plasma to the material. So, this assay is very useful, it tells you whether the complements will get activated. So, we have the previous coagulation based assay or hemolysis based assay, which tells you whether the blood the plasma going to get damaged, whether the biomaterial is going to activate your thrombosis and the coagulation pathway, and then comes whether the biomaterial is going to activate your complement system, okay. So, all these are assays which tells you sometimes qualitative, sometimes quantitative the effect of the biomaterial on especially when they are coming in contact. So, these are in vitro assays. So, I can do it with cell lines, I can look at the gene expressions, I can take plasma, blood plasma, and then I can do experiments with the blood plasma and so on actually. So, these are all. Now comes the next step where you are taking the biomaterial to animals, that is called in vivo, to determine the biocompatibility and safety of the material in the biological environment. So, whatever we do in the lab, still it is not really biological. I can use different types of cell lines, I can take the blood plasma, okay, red blood cells, but still it is not really in vivo, but then when we take it to an animal, then it becomes in vivo. So, there are many guidelines for in vivo assessment of tissue compatibility. FDA has some guidelines, there are regulatory bodies such as ASTM, ISO, USP, they have many guidelines, okay, which tells you what are the types of tests you need to do when you are working with animals, okay, biological evaluation of medical devices, guidance on selection of tests, animal welfare requirements, because when we carry out experiments with animals we have to follow a lot of animal welfare guidelines, okay, and we have to abide by them. There are 19 guidelines for and specific test procedures, okay. So, all these guidelines we have to undertake, especially when we go into the animal studies. So, okay. So, we will look at these more in detail in the next class. Thank you very much for your time.